after we clone these two genes, we have the cDNA amino acid sequence. Then I need to decide what next, what's the next step. And then I feel like, like you climbing the mountain, you got to a certain place, you thought you are at the top of the mountain, but you look at it, it's still 500 miles up there. So when I get the cDNA, that's only 2 kb, and the gene is maybe 100 kb, so it's much bigger. So it has a very exponential amount of the work. So that's one direction, go to the gene structure. And then you can also go to the direction looking at the structure and function that you make mutation to see which active site. Or you can go to the promoter region to study how is this gene regulated. So then you can also make the knockout mice using the gene. So I need to decide which direction to go. And it all sounds so interesting to me, so it's very hard for me to make decision. And of course, you are limited by the man-woman power in the lab, the funding, the time, so you have to choose one direction to go. So I thought, so at that time I go two directions. One is structure function, the other is a gene structure. So when we found the gene structure, that was a very, very happy experience because I have one student work on A gene and another student work on the B gene. That's a PhD project for each student. And one day I put these two genes together. The intron-exon organization was identical. I feel like I got the lottery because all the number matches. And so we found that they have an identical intron-exon organization. That suggests that they are coming from same ancestor gene million years ago. That is an important finding and has a lot of implication to evolution as well.